We know that DNA code stores the information needed to make every protein or protein enzyme the body needs to build and repair itself. And if DNA is the archive of information, then RNA is the courier that carries that information to the ribosomes where the proteins are built. In general, there are three types of RNA: rRNA, mRNA, and tRNA. And these RNA have three different functions. Ribosomal RNA, or rRNA, carries instruction on how to construct ribosomes, the protein factories in a cell. Much of the ribosome is composed of rRNA, along with ribosomal proteins. Messenger RNA, or mRNA, carries the information on how to build proteins from the DNA in the nucleus out to the ribosomes in the cell cytoplasm or attached to the rough endoplasmic reticulum. Transfer RNA, or tRNA, picks up free-floating amino acids in the cytoplasm and carries them to the ribosomes, where the amino acids will be assembled into proteins per instructions carried by the mRNA. There are many other types of RNA and RNA functions, but rRNA, mRNA, and tRNA are the most common types of RNA encountered in a cell. Unlike the double helix structure of DNA, an RNA strand is formed as a single spiral. RNA is read and copied by RNA polymerase transcription. The RNA polymerase enzyme reads one strand of the DNA molecule and then synthesizes the RNA strand one nucleotide at a time. The RNA polymerase reads the DNA in the 3' to 5' direction and assembles the RNA strand by fusing each nucleotide from its 5' phosphate onto the 3' OH group onto the initial RNA nucleotide. Of course, we remember the RNA nucleobases are adenine A, guanine G, cytosine C, and uracil U. The nucleobases pair with the DNA bases, GC and CG. The difference between DNA and RNA is that RNA uses uracil instead of thymine, so instead of the adenine pairing with thymine, adenine pairs with uracil, so we get AU and UA pairs with RNA. The purpose of RNA transcription is to retrieve protein-building information stored on the DNA strands. In prokaryotic cells, all the transcription for rRNA, mRNA, and tRNA is done by a single enzyme called the RNA polymerase holoenzyme. This holoenzyme has two parts, the core and the sigma transcription factor. The sigma transcription factor seeks out certain sequences of code at the beginning of a gene called a promoter region of AGCT code. The sigma transcription factor latches onto that promoter region. From there, the core polymerase breaks away from the sigma transcription factor. The core polymerase split open the DNA strand into two separate strands, the template strand and the coding strand. On the template strand, the deoxyribose sugars on the nucleotides are oriented in the 3' carbon to 5' carbon direction headed away from the promoter. The polymerase begins reading the DNA sequence in the 3' to 5' direction on the template strand with the 3' to 5' orientation. As it reads, the core polymerase assembles complementary RNA nucleotides in the 5' to 3' direction on the new RNA strand it builds from those complementary RNA nucleotides. If we remember from DNA synthesis, complementary RNA coding means the complements for AGCT on the DNA strand are UCGA for the RNA strand. The RNA polymerase knows to stop reading and transcribing when it hits a long sequence of repeating code. In row termination, a row protein climbs the RNA and simultaneously cuts the RNA and knocks away the RNA polymerase holoenzyme. Row termination is illustrated in the figure. In loop termination, just before the repeating stop sequence, the DNA code has a repeating group of GGCC followed by random code and then an inverted repeating code of CCGG. The transcribed RNA will complement the inverted repeating groups with its own complementary sequence CCGG random GGCC. The G's and C's nucleotides will attract each other and triple hydrogen bond into a loop. The loop will knock away the polymerase holoenzyme, and proteins will come in and cut the loop to free the RNA strand. In both termination cases, with the polymerase gone, the two DNA strands will pull together and bond back together in their original anti-complementary sequence double helix. In eukaryotic cells, there are different RNA polymerase enzymes for each type of RNA, and general transcription factors that help the RNA polymerase find the promoter regions on the DNA and help the RNA polymerase bind onto the DNA to start reading and transcribing the DNA code into RNA. In eukaryotic cells, the promoter regions might have one of three code sequences: 1 TATA, 2 CAAT, 3 GC. These sequences are called initiation signals. When general transcription factors come across initiation signals that matches its own initiation code, it knows it is at the correct promoter region. The general transcription factor binds to this promoter region on the DNA so that the RNA polymerase enzyme can start its RNA transcription work, as shown in the figure. An elongation step follows. In eukaryotes, termination of the RNA transcription occurs when the RNA polymerase transcribes the polyadenylation sequence AAUAAA onto the new RNA strand. This AAUAA is the termination sequence. This sequence triggers termination proteins, including endonuclease, to come in and cut the RNA strand at AUAA and knock away the RNA polymerase. The polyadenylation sequence AAUAA becomes the end of the RNA strand. Eukaryote RNA transcription and termination is shown in the figure. Special transcription factors enhance or depress the rate of transcription. These also have coded signals on the DNA, so that the special transcription factor will know where they should bind to the DNA strand. For ribosomal RNA, or rRNA, it takes a general transcription factor to bind the RNA polymerase type 1 to the DNA. The RNA polymerase 1 will read the DNA from 3' to 5' and use complementary RNA nucleotides to construct the new rRNA. The complete strand of rRNA will break away and be transported to the nucleolus body within the nucleus, where the rRNA will be incorporated into the structure of a ribosome protein factory. The ribosome will leave the nucleus through a nuclear pore in the nuclear envelope and attach itself to the rough endoplasmic reticulum, where it can begin making protein. For messenger RNA, or mRNA, the transcribing enzyme is RNA polymerase type 2. It also takes a general transcription factor to bind the RNA polymerase type 2 to the DNA. The RNA polymerase 2 will read the DNA from 3' to 5' and use complementary RNA nucleotides to construct new mRNA. This new RNA sequence is called pre-mRNA. The pre-mRNA must be modified and cleaned up before becoming the mRNA that can be used to make protein. I will discuss the pre-mRNA cleanup process in due time. After encountering the termination sequence AUAAA, the pre-mRNA will be cut away by endonuclease and be transported from the nucleus through a nuclear pore in the envelope. In the cytoplasm, the pre-mRNA will be modified into mRNA. The details of the modification will require a lengthy explanation. The mature mRNA will seek out a ribosome and bind itself to that ribosome. The ribosome will use that mRNA as a template to build peptide chains from amino acids brought into the ribosome by strands of tRNA. The peptide chains will be linked and folded into the protein that the DNA back in the nucleus provided mRNA the amino acid code to build. Besides mRNA, the RNA polymerase type 2 also makes snRNA, or small nuclear RNA, that is used in modifying pre-RNA into mature mRNA. The snRNA is used in splicing the mRNA back together after it has been modified from pre-mRNA. For transfer RNA, or tRNA, the transcribing enzyme is RNA polymerase type 3. It also takes a general transcription factor to bind the RNA polymerase type 3 to the DNA. Like type 2 polymerase, RNA polymerase type 3 also makes snRNA, or small nuclear RNA, that is used in splicing pre-RNA into mature RNA. Back to mRNA. The RNA polymerase II will read the DNA from 3' to 5' and use complementary RNA nucleotides to construct new mRNA. After some modification, the complete strand of mRNA will break away and will leave the nucleus through a nuclear pore in the envelope. The cleanup and modification of pre-mRNA into mature mRNA requires more discussion. Technically, the official name for pre-mRNA is heterogeneous nuclear RNA. But I'm going to continue to call it pre-mRNA because the name heterogeneous nuclear RNA is too cumbersome. To start the cleanup and modification of the pre-mRNA, we focus on the ends of the pre-mRNA strand. We know the 3' OH end or termination end of the pre-mRNA ends with termination signal A, a UAA. A poly-A polymerase enzyme will recognize this A, U, a termination signal, and add on hundreds more adenosine A nucleotides. The extra A's at the 3' OH end of the pre-mRNA is called the poly-A tail. The poly-A tail will protect the end of the RNA from erosion by nuclease enzymes. The poly-A tail will help carry the RNA from the nucleus out through a nuclear pore and then to a ribosome. The ribosome will be able to recognize the 3' end of the mRNA from its poly-A tail. The head of the pre-mRNA begins at the 5' end. The 5' end starts with 3 phosphate groups, PPP. In the first part of modification, a RNA triphosphate enzyme comes in and cleaves away one phosphate. So the 5' end of the pre-mRNA starts with 2 phosphates, PP. Next, a guanylyl transferase enzyme comes in and bonds a GTP, guanosine triphosphate, to the 5' end. The bonding of the GTP to the 5' end of the pre-mRNA releases 2 phosphates. So the 5' end of the pre-mRNA now starts with 2 phosphates and a GMP, guanosine monophosphate. Next, a methyl transferase enzyme comes in and adds a methyl group, CH3, to the GMP in a process called capping. The 5' end of the pre-mRNA is now said to be capped by a 7-mg or a 7-methyl-guanosine group. The 7-mg capping prevents nuclease enzymes from coming in and breaking down the pre-mRNA into nucleotides. The 7-mg capping also serves as a signal for the ribosome that this is the end where protein translation should be initiated. The next process in cleaning up and modifying the pre-mRNA into the mature mRNA is the cutting and splicing of introns and exons. Not all the transcribed pre-mRNAs code for amino acids. The 7-mg cap and the poly-A tail are just markers for the ribosome where to start and where to finish protein coding. There might be a few non-coding regions at the cap and the tail end of the pre-mRNA referred to as the leader and trailer nucleotides. The body of the pre-mRNA contains exons and introns. The exons are sequences that code for amino acids. The introns are sequences that do not code for amino acids. The introns must be cut out of the pre-mRNA. Once the exons are spliced back into the RNA strand, it can be called a mature mRNA, and it is ready to be used by the ribosome. The exons and introns are shown in the figures. The discarded introns are broken down by nuclease enzymes into nucleotides that are free to be used in more RNA transcription. Introns are not junk DNA. Introns provide diversity to mix and match, and so create new proteins and genes. A rule of population genetics is that large populations will weed out introns, but small populations will tend to retain and fix in place introns as a normal part of the genome. At first, introns were a useless result of endosymbiosis with bacteria, but some introns have become necessary gene regulators or gene enhancers and so are retained. Now the time comes for the small nuclear RNA, the snRNA, which are also called spliceomes. The introns are cut out of the pre-mRNA by the small nuclear RNA, snRNA, transcribed by the polymerase II or polymerase III. The snRNA must be combined with ribonuclear proteins to form SNRPs. It is the SNRPs that cut the intron from the exons. The 5' end of the intron marked by GU sequence of nucleotides. The 3' end of the intron is marked by a AG sequence of nucleotides. Somewhere in the middle of the intron is a branch point adenine A, or an adenosine nucleotide that a SNRP will use as an anchor point. One SNRP finds the GU sequence and cuts the intron from the first exon, exon 1. This action will expose the OH at the 3' OH end of exon 1. The SNRPs pull the cut GU end of the intron around into a loop and bond the GU to the A branch point. Another SNRP will cut the intron loop away at the AG intron at its 3' end and expose the phosphate at the 5' PO4 end of the second exon, exon 2. The exposed OH on the exon 1 will attract the exposed phosphate of exon 2, and exon 1 and exon 2 will be spliced back together with a phosphodiester bond. This process of the SNRPs cutting out introns and splicing the exons back together will continue until all the introns have been excised from the pre-mRNA. Once all the exons have been spliced back together, we can now call the mRNA a mature RNA, ready to be brought into a ribosome to make proteins. The whole SNRP intron exon cutting and splicing process is shown in the figure. Two variations on the straight conversion of pre-mRNA into mature RNA are alternative RNA splicing and RNA editing. Both variations require all the introns to be removed. Any introns left in a mature RNA strand might result in a distorted protein that does not function or performs a malicious function. In alternative RNA splicing, some exons might be left out. The result is a variation of the full protein that would be formed by the full mature RNA. These variations still form functional proteins that might have a slightly different effect than the full exon protein. If the effect is beneficial, it is retained. An example of beneficial variation might be a slightly different antibody protein that might kill a mutated virus better than the full antibody protein. Another example of a beneficial variation might be a slightly different protein that puts a chemical receptor in a slightly different place that result in better reception of a chemical or a reception of a variation of the chemical. Obviously, a major variation from the full protein might cause loss of the protein's function. An illustration of alternative splicing is shown in the figure. In RNA editing, a change might be made by an enzyme to change the stopping point of protein translation. For example, there might be a full protein that affects the function of blood cells. If that same RNA is produced in an intestine wall cell, it might not need to produce all the effects it produces for blood cells, and the protein could be a shorter variation of the full protein. The stop codons for amino acid assembly are UGA, UAA, and UAG. If translator polymerase comes across any one of these stop codons, it stops adding amino acids to the protein it is assembling. The mature RNA might have a codon variation CAA, the C standing for the nucleobase cytosine. The difference between cytosine and the nucleobase uracil is cytosine has amino group NH3 in the same places that uracil has a carboxyl group CO. If a shorter variation of a protein is desired, a cytosine deaminase enzyme can come in, replace the amino group NH3 on the cytosine with a carboxyl group CO of uracil. The result would be the amino acid codon CAA becomes the stop codon UAA. The translation polymerase in the ribosome would see the stop codon UAA and stop adding amino acids to the protein. The result is a shorter protein that might do a better function in an intestine cell than a blood cell. In this way, one gene can produce two variations of a protein for two different cells with two different functions. It must be by some other bit of information that the cytosine aminase enzyme knows when to act and when to be absent. Illustrations of RNA editing are shown in the figure. In preparation of tRNA and rRNA, intron cutting and exon splicing still might be needed. However, where intron cutting and exon splicing is common in mRNA preparation, intron cutting and exon splicing is rare in the preparation of tRNA and rRNA. Introns are common in most eukaryote DNA. Introns do not exist in prokaryote bacteria. In bacteria, DNA is transcribed directly into mRNA, without the pre-mRNA intron exon splicing that occurs in eukaryote mRNA preparation. Prokaryotes, bacteria, and archaea can repair their own DNA that has been infected or damaged by viruses. By replacing the damaged sections with CRISPR, clustered regularly interspaced short palindromic repeats of nucleotides. By studying how bacteria repair their DNA using CRISPR, scientists have developed CRISPR techniques of splicing in genetically engineered sections of DNA code into both prokaryote and eukaryote DNA, including mammalian and human DNA. The CRISPR gene editing technique has been used by geneticists to genetically modify proteins and organisms in the laboratory, to make man-made or man-altered proteins and organisms. Other forms of RNA besides mRNA can also be transcribed directly from DNA. Transfer RNA or tRNA is transcribed from DNA in a similar fashion to mRNA transcription. There may or may not be the same intron cutting and exon splicing process with tRNA as occurs with mRNA. Mature mRNA is often several hundred to several thousand nucleotides long. Conversely, tRNA is only 80 or so nucleotides long, so tRNA is a lot shorter than mRNA. The shortness of tRNA cuts down the likelihood that there will be random intron sequences in the tRNA. So tRNA can usually be transcribed directly from the DNA without all the intron exon cutting and splicing of mRNA. The function of tRNA is to gather amino acids in the cell's cytoplasm and carry them to the ribosome. Every tRNA has a 3-letter anticodon that will be specific to an amino acid. It is the ribosome that assembles the amino acids into peptide chains that will become proteins. Labeled representations of a tRNA molecule are shown in the figure. The key parts of tRNA are the D loop, the T loop, the variable loop, the anticodon loop, and the stem at a 3' end of the tRNA with its CCA tail sequence. The D loop holds the amino acyl tRNA synthetase enzyme that reads the anticodon sequence, identifies matching amino acid to the anticodon, and uses an ATP to attach that amino acid to the 3' CCA tail of the tRNA. The T loop tethers the tRNA to the ribosome. The variable loop plays a role in helping the tRNA synthase recognize amino acids when necessary. The variable loop may be absent when its amino acid recognition role is not necessary. The figure shows 2 representations of transfer RNA. The left figure emphasizes the nucleotides. The right figure emphasizes how the anticodon on the tRNA complements the codon on the mRNA. Note the amino acid on top of the CCA tail. The anticodon loop is the most important loop on the tRNA structure. The anticodon loop has a triplet of nucleotides that are complementary to the codon of the messenger RNA, to which it will key onto. Inside the ribosome, the mRNA is laid out on the ribosome small subunit. The ribosome large subunit will allow the incoming tRNA with a bound amino acid to enter. If the tRNA anticodon is complementary to the codon of the mRNA, the ribosome enzymes will release the tRNA-bound amino acid and add that amino acid to the peptide chain that will become the protein. Once the amino acid becomes part of the peptide chain, the ribosome releases the empty tRNA to seek out another amino acid that matches its anticodon. We will discuss the protein coding of mRNA in more detail in the next section. For anyone with a professional interest in genetics or medicine, Professor Zach Murphy gives a more detailed explanation of RNA transcription and protein translation on the YouTube channel Ninja Nerd at the link shown. In my next video, I will discuss codons and the genetic code.
Thank you for watching. If you enjoyed this video and want to help me make similar content, please like and subscribe.